The overall goal of this procedure is to get skills and knowledge to isolate DNA and RNA by cell lysis, protein removal, and precipitation to analyze the integrity and amount obtained by spectrophotometry and electrophoresis.
For DNA isolation, it is important to use RNases in the lysis process in order to degrade RNA present in the sample besides using a detergent like SDS. While protein removal, it is important to add phenol at a neutral or alkaline pH such as A to maintain the nucleic acids in the liquid phase. For the RNA isolation, it is important to maintain the sample free of RNases and to use DNases. It is important to add phenol at an acid pH such as 5.
The cellular lysis will vary depending on the nature of its envelope. In the case of Lactobacillus sp, we have a gram-positive bacteria composed of a cell wall made of a thick peptidoglycan layer that surrounds a cytoplasmatic membrane. In the case of an Arabidopsis thalia, we have a cellulose wall, and in the Homo sapiens saliva there is only a cytoplasmatic membrane. For Lactobacillus sp, it is used one ammonia detergent to degrade cell membrane, lysozyme for the cell wall. For Arabidopsis thalia, liquid nitrogen, dry ice. For Homo sapiens saliva, lysis buffer with trees, EDTA, sodium chloride, and SDS. To eliminate proteins, phenol, and chloroform, proteinase K can be used, or chelating agents such as EDTA. To precipitate and recover DNA with a mixture of cTA and sodium chloride in high concentrations. Prefer to use sodium acetate because it works as a buffer. Ethanol at 70%.
The most important DNA isolation techniques are solid phase-based kits, columns with phenol, chloroform extraction, and monophasic reagents. It's based on the selective binding of DNA to silica-based membrane in the presence of chaotropic salts. There are four methodologies that allow us to quantify nucleic acids. We have spectrophotometry, fluorometry, slot-bot, real-time PCR.
From human blood, a tube with blood was shaken by immersion several times to ensure smooth transferring, and a liquid of 4.7 milliliters in a microtube was transferred. The volume was adjusted to 1.5 milliliters with sterile water, centrifuging at maximum speed, 13,500 RPMs for 1 minute. 1 milliliter of supernatant was discarded, being careful not to break up the pellet containing the leukocytes. 1 milliliter of sterile water was added and mixed thoroughly by immersion and centrifuged at 13,500 RPMs for 1 minute. Supernatant was discarded, taking care of not breaking up the pellet. 375 microliters of 0.2 molar sodium acetate pH 7, 25 microliters of 10% SDS, and 15 microliters of proteinase K solution were added. The pellet was resuspended. 37 degrees was incubated. It was centrifuged briefly. 200 microliters of FCA 25-24-1 were added and stirred for 20 seconds until an emulsion was obtained. It was centrifuged, recovering the aqueous phase into a new tube. The phases took off, so we centrifugated it again 3 minutes. 1 milliliter of cold absolute ethanol was added and stirred slowly for immersion. It was centrifuged. The supernatant was removed and the pellet was resuspended in 200 microliters of 0.2 modium sodium acetate, incubated at about 55 degrees for 10 minutes. 500 microliters of absolute ethanol were added, stirred, and centrifuged. Washing was repeated with 1 milliliter of 70% ethanol, and the pellet was left to dry in the open tube upside down on absorbent paper. The pellet was resuspended in 40 microliters of water. Level molecular biology. It was properly labeled and stored at minus 20 degrees.
A culture of bacteria E. coli top 10 was run overnight, 16 hours in Loria Bertani at 12,000 RPM for 1 minute. It was centrifuged, 1.5 milliliters of bacterial culture in a microtube. Then supernatant was eliminated. This was made twice. The tablet of bacteria was resuspended with 600 milliliters of lysis buffer test. It was incubated 10 minutes at 37 Celsius degrees and left at room temperature for 1 minute. 800 microliters of phenol were added, mixed well by immersion and incubated at room temperature for 5 minutes. 160 microliters of chloroform were added, mixed thoroughly by immersion. It was centrifuged. The aqueous phase was transferred to a new microtube. 0.8 volume of isopropanol were added, mixed by immersion and incubated at room temperature for 10 minutes. Centrifuge. The supernatant was discarded, taking care not to break up the RNA pellet. 1 milliliter of 70% ethanol was added and mixed by immersion several times, taking care not to disgregate the RNA pellet. The tube was placed upside down on paper towels at room temperature until dry. The tablet pellet was resuspended in 30 microliters of molecular biology grade water. The tube was labeled approximately at and stored at minus 20 Celsius degrees. Concentration measurement by spectrophotometry. Make a dilution of 1 to 100 of each sample DNA RNA with double distilled water. Make a final volume 1 milliliter. Turn on the spectrophotometer and set the wavelength of 260 nanometers. Place in a cell for spectrophotometer 1 milliliter of water BM and read it as a target. digit. Remove the blank and insert the dilution of our DNA with respect of RNA. Read the sample and record the result. Take readings of quality index and concentration. Prepare a gel 0.8% agarose. Load samples premixed with loading buffer in the following order. Marker 1 kilobase plus 5 microliters. Human genomic DNA 5 microliters. RNA of bacteria 5 microliters. Spread the gel at 120 volts with TBA running buffer.
Quantification by spectrophotometry using the absorbance coefficient showed a value of 11.0063 micrograms per milliliter for the DNA dissolution, which indicates a concentration of 1100.63 nanograms per microliter, recovered in 30 microliters. For the RNA dilution, the value was 1.1796 micrograms per milliliter, which indicates a concentration of 117.96 nanograms per microliter, recovered in 40 microliters. This means that 33,018.9 micrograms of DNA were deposited in well 1 and 4,718.4 micrograms of RNA in well 2.
In order to talk about purity and protein contamination, the quality index was used, using an absorbance of 260 for DNA and 280 for proteins. For DNA, it was obtained a value of 1.7327 and for RNA of 1.2317. The electrophoresis shows the migration of the DNA ladder by oxygen in well 1, the DNA sample at well 2, and the RNA sample at well 3. It can be seen that at well 2 there is degraded DNA, protein contamination, and a low rate of migration. At well 3 it can be seen DNA contamination, protein contamination, salt contamination, and degraded RNA at the bottom.
It was obtained a quality index of 1.2317 for the RNA sample at the spectrophotometer, which indicates a low purity talking about protein contamination. An appropriate value must oscillate between 1.7 and 2, which indicates that even though we have more content of RNA than that one of proteins, it is not pure. For the DNA sample, the quality value obtained was of 1.7327, which indicates an acceptable purity to work, but it must be confirmed with electrophoresis.
In the agarose gel, we can confirm the contamination of proteins because there was sample start in the wall. Also, we can see degraded DNA contamination and salt contamination. For the DNA, it was expected an acceptable quality of the sample according to the results of the spectrophotometry, but unfortunately, we weren't exempt of protein contamination. For PCR sequencing and the usage of restriction enzymes, when we have higher levels of proteins, that ones could inhibit enzymatic activity and also hindered by the presence of other proteins would lower the yield of the reaction. If there is contamination with salts, that can also encourage the start activity.
When isolation DNA or RNA, it is important to consider the type of cell that will be used in order to use the technique that best suits the lysis, the proteinization, and precipitation of the nucleic acid. It is important to use a target that can let us measure the absorbance with the spectrophotometer and to select the correct wavelength to quantify the appropriate type of mold. For DNA and RNA visualization in an agarose gel, it is important to select the agarose concentration that best fits the sample size if it is known. It is also essential to consider the running conditions according to the time and distance between electrodes. Protein, salts, etc. may appear in the gel, and it is important to visualize this in order to determine if it is necessary or not to repurify the sample.